Today we are going to demonstrate the gram stain technique, which has been around since the 19th century after the invention by Danish scientist Hans Christian Gram. This gram staining technique divides the bacteria into two different groups, which is two broad categories: gram stain positive and gram negative bacteria. Gram-positive bacteria has peptidoglycan cell wall and stains purple after the staining procedure, whereas gram-negative bacteria does not have peptidoglycan cell wall and stains pink after the staining procedure. For this staining procedure, the materials needed are inoculating glue, incinerator, DI water, biblious paper, a microscopic slide, a tray to do the gram staining, and the stains for the gram stains are crystal violet, iodine, alcohol as a decolorizer, and saffron. And last but not least, the bacteria to work with. We've already prepared a smear using aseptic technique. The best way to prepare a smear while using a solid medium is just touch the bacteria with the inoculating group and don't dig into the agar so that you don't get a clump of cells after the staining procedure under the microscope.
There is smear of bacteria, and this is crystal violet stain. It stains everything that's on the slide, like the protein and cells of the bacteria. It has to remain on the slide for 10 to 60 seconds. After 10 seconds, we use the deionized water. Just let the water run over the slide and be careful not to wash off the bacteria from the slide. This is gram iodine. It complexes the crystal violet stain. It has to remain on the slide for 10 to 60 seconds as well. After 10 seconds at least, we use deionized water again to wash it off. This is gram decolorizer. It decolorizes and removes crystal violet stain. It has to remain on the slide for just 10 seconds. Be careful not to place the stain for more than 10 seconds. So just wash it off quickly with deionized water. If the bacteria has peptidoglycan cell wall, it retains the crystal violet and turns purple. If the bacteria does not have the peptidoglycan cell wall, it does not retain the crystal violet and does not turn purple. This is the decolorizer that we just used. This is the saffron. That's the counter stain. It stains everything that's been decolorized by the decolorizer. It has to remain on the slide for 10 to 60 seconds. And after 10 seconds, we use deionized water to wash it off. After the completion of the staining procedure, we just use the biblious paper to blot the slide. Place the slide inside the paper and blot it gently. And slide is ready to view under the microscope now.
This is a microscopic view of the bacteria after the gram staining process. The gram-positive bacteria stains purple, and we can also see the morphology, which is cocci, and the gram-negative bacteria stains pink, and the morphology of the gram-negative bacteria here is rod.